Hello, thank you very much for inviting me as keynote speaker here to this conference today. My name is Margit Tolle. I'm working at NPO Veggies Cultivation, and the title of my today's talk is The Influence of Effective Microorganisms on the Growth and Nutrient Content of Tomato Transplants. Presentation outline, short introduction. What is EM? What is Bokashi technology? Tomato transplants trials with EM, tomato transplants trials with Bokashi T. Effective microorganisms. The logo is on left hand side and how it is sold in the shops is on right hand side. EM1. This is EM1 not activated, but normally you use it in activated EM solution, which you can use in your garden and in plant production. EM technology was developed at the University of Ryukyus in Okinawa, Japan in the early 1980s by Professor Dr. Teruo Higa. EM technology has been introduced and it is used in more than 140 countries in the world. What is EM? EM is produced through a natural process of fermentation. EM is a combination of beneficial, naturally occurring microorganisms. EM is a liquid concentrate. EM is produced from cultivations of over 80 varieties of microorganisms. EM most outstanding feature is that it includes anaerobic and aerobic species of microorganisms coexisting in a most beneficially productive manner, not chemically synthesized or genetically engineered. It contains microorganisms from different groups. EM consists of safe and human-friendly microorganisms, which are main species of EM microorganisms: lactic acid bacteria, photosynthetic bacteria, yeast, actinomycetes, fermenting fungi. Coexistence. Lactic acid bacteria and yeast are producing organic acids, which are used by photosynthetic bacteria. Photosynthetic bacteria are producing vitamins and nucleic acids, which are used by lactic acid bacteria and yeast. The concept behind it. In soil normally are 5% of harmful microbes, 90% of opportunistic microbes, and 5% of beneficial microbes. What happens when we add EM to the soil? Then opportunistic microbes are going to work as beneficial microbes, and this is shifting the balance to suppress disease. EM technology for agriculture. EM technology increase yield and improve the quality of production. Organic waste can be converted into fertilizer, soil improvement. EM is there are two types of EM, liquid type and solid type. Liquid type are EMAS and EM5. EMAS, EM activated solution, is used for crop production, for increasing yield and crop quality, and EM5 is meant for crop protection, to protect plants against pests and diseases. Solid type. EM Bokashi aerobic, EM Bokashi anaerobic, EM compost, EM mothball. EM1 can be activated and multiplied for making EM-related materials. Differences between EM1 and EMAS, which is EM-activated solution. EM1 quality is excellent, activity is steady, storage 6 months. EMAS, EM-activated solution. Quality is good, activity is high, storage 1 month. EM bokashi, anaerobic EM bokashi in tight containers, and aerobic EM bokashi where air is around the bokashi. EM mothball. EM mothballs can be used for wastewater treatment, especially for sludge treatment. It can reach the bottom and remains longer than EMAs. Beneficial influences of EM. EM promotes germination, flowering, fruiting and ripening in plants. EM improves the physical, chemical and biological environments of the soil and suppress soil-borne pathogens and pests. EM enhance the photosynthetic capacity of crops. EM ensure better germination and plant establishment. EM increase the efficiency of organic matter as a fertilizer. Different brands of EM are applied as follows. Inoculation of EM into the soil, spraying of EM on leaves, soaking seeds in EM, EM irrigation fertigation, insect control by EM5. Tomato transplants experiments with EM. Problem: elongated, low-quality tomato variety valve transplants. When I started this experiment, I was looking for problems in tomato cultivation. And then I heard from an agronomist that there is a tomato variety valve, which tomato transplants are elongated and in low quality when transplanting, and then I decided to take this variety and see what EM can do with this variety. And the aim was the purpose of this investigation was to assess the influence of effective microorganisms on the growth and nutrient content of tomato transplants. Treatments: EM treatment and control treatment. Seed soaking half an hour in EM solution, EM treatment, and in water control. Substrate treatment: EM 1 to 500 solution and water in control treatment. Third through leaf stage spraying: EM 1 to 1000 solution and water as control treatment. 1 week later, EM to 1000 solution and water in control treatment. And again 1 week later, EM 1 to 1000 solution in EM treatment and water in control treatment. And again 1 week later, EM to 1 to 1000 solution and water in control treatment. And here you can see the results of the experiment. Tomato transplants were lower in EM treatment than in control treatment, and it was about 30% lower, so the results were remarkable in the height of transplants. The width of tomato transplants. Here you can see that the results are not statistically different. The number of leaves per tomato transplants, here you can see that the results are not statistically different. And stem diameter of tomato transplants, here you can see that the stem diameter was increased uh, compared to control treatment, and it was about 25%, so the results were remarkable. The content of nutrients of tomato transplants. Here you can see that the content of nitrates in tomato transplants was higher in EM treatments than in control treatment. And this is not a good thing, as you all know. The content of nitrogen was also higher in EM treatment compared to control treatment. The content of phosphorus was higher in EM treatment compared to control treatment. And the content of potassium was higher in EM treatment than in control treatment. The content of calcium was higher in EM treatment than in control treatment. And the content of magnesium was higher in EM treatments than in control treatment. And you know that this is a very rare case when you get all analyzed nutrients higher in analyzed treatment, EM treatments, and in control treatment. Normally it doesn't happen, this picture. And you can see the transplant of tomato at the end of experiment, which is in EM treatment. Photo of tomato transplants. On the left you can see EM treated plant, and on the right you can see control plant. And you can see that the control plant is elongated and few leaves on the stem, and the EM treated plant is with higher stem diameter, and the plant is shorter. And why it is important? It is important because when plant is shorter you get quicker plant nutrients to the top of the plant, and when plant has larger stem diameter you can get more nutrients at the same time going up to the plants. Effective microorganisms improve the quality of tomato transplants because they stay compact and stem diameter was greater. Photo of the same tomato plants which you saw in last picture after the season, right at the ending the season, in the end of September in Estonia. And you can see that the control plant is almost finishing the growing season already, and few uh, tomatoes and with very small size. And you can see that the EM treated plant has more living power, is in full growing yet, and has larger tomatoes on it. Bokashi technology experiments. Introduction. Bokashi substrate such as rice bran or wheat bran is inoculated with a mixture of beneficial microbes, effective microorganisms that flourish in aerobic and acidic environments. Bokashi fermentation holds several advantages over traditional composting. The process is performed in a closed and anaerobic system. It does not require mechanical intervention, preventing the loss of nutrients and greenhouse gases efflux. The acidic and anaerobic environment quickly suppress the viability of pathogens. Using a selected microbiome as a starter, the process minimizes the production of putrid odors. This fermentation breaks down the complex organic carbon into short chain fatty acids, which are readily available nutrients for animals, soil microorganisms, and plants. The prepared EM bokashi fermentate is thus a compound product containing organic matters at a range of decomposition stages, high biomass of a specific microbial community, and the metabolites. Here you can see on the left-hand side bokashi in a container, and on the right-hand side the grate in the bottom of the container. Here you can see on the left-hand side the food waste in the container, and on the right-hand side the top on the container to collect bokashi tea, which is a liquid fertilizer. Here you can see on the left-hand side the container closer provides an anaerobic environment, and on the right-hand side powerful healthy plants growing on the compost containing bokashi fermented product. And the aim of the experiments was to see how the bokashi tea treatment influences the tomato transplant growth and performance. And the treatments were bokashi tea treatment and control treatment. Seeds were soaking in half an hour in EM 1 to 500 solution in bokashi tea treatment, and with water in control treatment. Substrate treatment was in bokashi tea treatment with EM 1 to 500 solution and with water in control treatment. Spraying from the stage of third through leaf stage with bokashi tea treatment with EM 1 to 500 solution and with water in control treatment. 1 week later with EM 1 to 1000 solution and with water in control treatment. And again 1 week later EM 1 to 1000 solution spraying in bokashi tea treatment and with water spraying in control treatment. What are the results? The results are that the plant height, the number of leaves, and the stem diameter of tomato transplants can be seen here. The plant height was higher in bokashi tea treatment than in control. The number of leaves were not statistically different, and the stem diameter was higher in bokashi tea treatment than in control treatment. Bokashi tea treated plant on the left hand side and right hand side control plant. And here you can see that the plant is higher, the stem diameter is larger, and here you can see some books what I wrote, and you can find them on this website www.veggiescultivation.com/books, and there are 12 research monographs in English. Here is methods to avoid calcium deficiency on greenhouse grown leafy crops. Effective microorganisms influences vegetables and soybeans production. Silicon as a challenge for sustainable vegetable crop production. Light and vegetable production. Advances in tomato cultivation. Vermicomposting is boosting greenhouse vegetable production. Successful legume cultivation in northern hemisphere, organic cultivation and vegetables and potatoes, innovative technologies for sustainable crop production, best effective microorganism practices in greenhouse plant production, best practices in greenhouse plant production, and best practices against calcium deficiency in greenhouse plant production. And three popular scientific books, two in English, Bokashi Digested Bokashi Technology, The Best Way to Save Our Planet Earth in EM Technology, and one in Estonian, Vegetables for Garden. Acknowledgements. Thailand Saraburi Farm EM Seminar Materials. The project Environmentally Friendly Vegetable Production Development Results project was supported by Estonian Agricultural Registers and Information Board, Jaagumäe Agro Ltd and Estonian Crop Research Institute. Right Farm Experiences, collaboration with Kõlleste Garlic, Margit-Olle other scientific work materials and other PowerPoint presentations and also my article. And here you can see again the best research results plants, the tomato transplants in EM treatment on the left hand side and tomato transplants in control treatment on the right hand side. And with that, I thank you for listening to me. And unfortunately, I cannot be today here to answer all of your questions, but I wish you successful conference. Further on. Thank you very much.